All right, so now let's go ahead and get into the cell-mediated immunity. And this is where we're getting into our CD4 and CD8 cells. CD4 are the helper T cells. These guys help by secreting cytokines. Basically, they're going to help moderate the immune response. So they're basically, a lot of other immune cells can't do anything, or they can't really activate truly unless the CD4 T cell is there. This is also the target of HIV, and so like that's why HIV can be so catastrophic, is because other immune cells are, if the CD4 cells are gone, other immune cells don't have basically a boss to tell them what to do. So CD4 helper T cells, they're going to be secreting these cytokines, secreting these signals to try to moderate the process of the immune response. Now CD8 cells, these are the killers. They're going to find those cells that are infected or cancerous cells, right, whatever, and they're going to kill those cells off. So if a cell, basically, like if a cell becomes infected with a virus, it's basically a zombie, and the CD8 cell is going to be the zombie killer, right? Cell-mediated immunity, or CMI, this is really effective against intracellular pathogens, basically things that can get into a cell and take over that cell. So fungi, parasites, viruses, cancerous cells, right? It also is going to be what is going to target tissue transplants, and that's why people who get like an organ transplant need to take immunosuppressive drugs. It's because these cells are going to be triggered against it. So what do we do? What's the steps to get this cell-mediated immune response going? Number one, the antigen has to be presented, right, either by an MHC, or a major histocompatibility molecule, on an antigen-presenting cell, right? This is like a dendritic cell. This will be recognized by the CD8 or the CD4 cells. Some of those T cells will proliferate, meaning they'll copy themselves, activate, and then differentiate into basically a bunch of clones. And a lot of those are going to be effector cells, because that's what we need in that moment to fight the infection, with a handful of memory cells so we know how to fight it later on. Like I mentioned in the previous video, once we finally get rid of the antigen, the infection is kind of over. The effector T cells are going to die down. The memory cells stay around. The memory cells are basically just waiting for us to come into contact with that same antigen again. So we look over here, what's going on in the picture. Here's our immature CD4 cell. Here's our antigen presenting cell. We have this major histocompatibility complex 2. It has the antigen on it, and it's presenting it to the CD4 cell, right? It binds to the receptor, and now we have a mature helper T cell, right, which can then release cytokines to help moderate immune responses. Over here, very similar, antigen presenting cell, except we have major histocompatibility complex 1, right, presenting that antigen to the immature CD8 cell, which then matures it. So now this cell can go after those specific antigen-containing cells. And you heard me mention in the previous slide, major histocompatibility complex. What the heck is that? So the major histocompatibility complex is also known as the human leukocyte-associated antigen, or HLA. So the HLA antigen. These are unique to you. This is unique to your immune system. There's a lot of different categories, or a lot of different versions of this, and your unique combination of them is what makes your immune system unique. And it also is why tissue transplants can be difficult, because everyone has a slight different combination of these antigens. So, but we can break it down into two classes, MHC1 and MHC2. MHC1 is going to present the antigen to the CD8 cells. And every cell except for red blood cells has these. So this is something that your CD8 cells can basically kind of go around and be like, all right, do you belong here or not? Right? So MHC cells, these are like, hey, every cell, for the most part, is going to have these antigens. MHC2 right? These are presented to the CD4 cells. And this is only going to come from the APCs, the antigen presenting cells. So only APCs are going to show the MHC class II antigens. Now, in order to present these antigens, we have to go through what's called antigen processing and presentation. We don't show the entire protein. We don't show the entire antigen. We're going to take that antigenic protein and we're going to break it down into specific short fragments, right, that are going to be combined with the MHC molecules and put onto the surface of the antigen presenting cell. So we're not presenting the entire antigen, just some specific portions, right? Just certain fragments are going to be presented. Right, so let's talk about MHC1 processing presentation. So this is going to be presented to CD8 cells. And again, pretty much any cell can do this, right? Pretty much any cell, with the exception of red blood cells. Basically, we're taking endogenous antigens. This is something from inside the cell. And these are going to be, again, antigens that are coming from like a virus. They're coming from a bacteria that's gotten into a cell. They're coming from antigens produced by a tumor, right? We break it down. We join the MHC1 and the antigen fragments together in the ER. We package it into a vesicle. We put it on the surface of the cell. Again, pretty much any cell can do this. MHC2, right? This is where we have an exogenous antigen. And the exogenous one, this is coming from outside the body. So the bacteria, a fungus, a protozoan, a free virus, something that has not gotten into a cell, right? It's an antigen from something that has not gotten into a cell. This is going to be processed and presented on this to the CD4 cells. And this MHC2 process is only going to happen on the antigen presenting cells like a dendritic cell. So in this case, we have to take the antigen in, because the antigen has been found outside of the cell. We have to take the antigen in, process it, right? Break it down like this. We make our MHC2 molecules, put those in a vesicle, join the vesicle with the peptide fragment vesicle. They bind, and then fuse that vesicle so we present it on the outside. So that's the process there. So they're very similar, with the main difference being MHC1 is dealing with something that's already gotten into a cell, and any cell pretty much can do this, whereas MHC2 is from antigens found outside the cell, and only antigen presenting cells can do this and use this. Those are the main things I'd like you to remember when it comes to MHC1 and MHC2. So now let's go ahead and get into how do we kickstart CD4 cells? How do we get these going? So this is where we have to activate and clonally select CD4 helper T cells. Right? So helper T cells or CD4 T cells, right? They're going to take the CD4 protein on the surface. They're going to recognize the antigen fragments on the MHC2 molecules. Because remember, MHC2 coming from an antigen presenting cell. They're going to recognize the antigen, and these helper T cells are then going to activate and start dividing, right? They're going to divide, right? We've been activated, right? And then create clones of effector cells and memory cells. And both of these memory and effector cells have the same receptors. The effector cells then are going to release the cytokines like interleukin-2, which is going to help trigger cytotoxic T cells and B cells. The memory cells, right, can hold on to this for long term. So let's take a peek at like what the helper T cell is doing. So we have this antigen presenting cell, in this case a macrophage, right? It has that MHC2 protein with the antigen. Interleukin-1 and other cytokines coming from the macrophage are going to help activate the helper T cell. when that binding is also going to help with the proliferation of it, of that helper T cell. And then by releasing things like interleukin 2 and other cytokines, we can trigger the activation of cytotoxic T cells as well as B cells, so that the cytotoxic T cells can go attack cells that have already been infected, or release things like interferon to protect cells that haven't been attacked yet, or activate the B cells, which will then secrete antibodies, right, to target the antigens. When it comes to activating CD8 cells, right, CD8 cells, right, they have the CD8 protein. They're going to recognize the antigen from MHC1. But recognizing that's not going to be enough, they also need some cytokines to help them, like interleukin 2, which is coming from the helper T cells. So they find the right antigen, they get activated, we proliferate, same thing, right? We get activated, we got to increase our numbers. Proliferate, differentiate, make effectors, and make memory cells. Again, they have the same receptor. The effectors are then going to go after and target cells that are infected using the perforin, using the granzymes, which will kill the cell that's been infected. Memory cells, as always, hold on to the information of how to target infected cells for that specific antigen. So I mentioned this before in one of the earlier videos, but we want to talk about it again because this is how a CD8 cell works. But these cytotoxic T cells, right, these CD8 cells, they are going after cells that have been infected or are cancerous, right? And they're going after these cells that have the same antigen, right, that goes with their receptor. So they can't just go after any and all, right? But they have a couple different ways of targeting and killing that cell. Perforin, which is going to basically punch a hole in the cell, which then causes fluid to just flood into the cell and burst it open. And granzymes, which will basically chew up the DNA and cause the cell to go through apoptosis, which is like a demolition, a controlled demolition of the cell. So we look over on the side, we see that, right? Here's our cytotoxic T cell, it's been activated, right? It's bound with the infected cell, right? Because the antigen and the receptor have come together. We're releasing perforin to punch holes. We see that it's bursting open from fluid flooding in. We have the granules that are going to go after the DNA and chew up the DNA. And then, again, just controlled demolition of the cell. Now, when it comes to B cells, we're working with antibodies, right? Antibody-mediated immunity. So the antigen, basically, we don't need to worry about an antigen-presenting cell here because the B cells can directly identify antigens. The antigen is actually going to be recognized and bind to the receptor on the B cell, and then that B, as well as the antibodies that have been secreted by the B cell, because remember, the antibody is just like the receptor, but in a free form. Now, helper T cells can co-stimulate B cells with the cytokine, so we can amp up B cell proliferation, so we can increase the numbers of the effector cells that can make more antibodies, and also produce the memory cells that hang around for a long time. And then also, when we get into contact with that same antigen, we can reactivate very quickly, because again, this is direct. So B cell, right? We have helper T cells that can use cytokines to help increase our numbers, and then the binding of that antigen to the B cell surface can also help trigger that as well. So when we activate a B cell, antigen binds to the receptor. Some of that antigen gets taken in, right? We use endocytosis, bring it in, we break it down, right? And then when we use our MHC2, we can put it on the surface, right? Because remember, MHC2 is going to be used by immune cells. So we put it on the surface, and then helper T cells are going to see the antigen and the MHC2 combo, right? They're going to deliver the cytokines like interleukin 2, 4, 5 to increase B cell proliferation, increase our numbers, right? So here we have, right, this microbe, it's bound, right, it has an antigen that's stuck to the receptor of a B cell, right? We've recognized the antigen, we go through clonal selection, but we can also have helper T cells that can co-stimulate it. Basically, the B cell has the processed antigen that is recognized by a helper T cell, which then releases the interleukins to also promote this. And then we go through, we make a bunch of B cell clones. Some of them are memory cells, some of them are plasma cells, where we can just produce antibodies like crazy. Same thing as we saw with the T cells. The effector cells, like the plasma cells, they're only going to be around while we need them. So we see high numbers when we actively have the antigen present, so we're actively infected. But then when the antigen is gone, plasma cells die back. Memory cells are the ones that generally hang around long term. So what's an antibody? What's the whole part, like what is an antibody? What does it look like? All that. So we know our B cells have this B cell receptor on the surface that is specific to a certain antigen. And we can send these receptors out free floating in the form of an antibody. They're antibodies, they're gamma globulins, immunoglobulins, right? These are sent out by the effector cells, the plasma cells, not the memory cells. The memory cells hold on to information on how to make them. The effector or the plasma B cells actually make them. So antibodies are basically the free-floating soluble receptors. They're found like this little Y shape, and we find them all over the place. Body fluids, secretions, all sorts. And there's 5 different classes or isotypes. I'm going to use the word classes. So 5 different classes: IgA, IgD, IgE, IgM, and IgG. Each of them have a different role, right? So they're all going to be similar, but they do different things when it comes to immunity. There's 2 key parts. There's the FC portion. This defines like the isotype, right? So which class does it belong to and whatever. And then the Fab region, this is for the specific antigen. So I know it's kind of a little complicated language here, but FC, this is the class. So think C for class. AB, antigens, antibody, right? That's how you can probably try to remember it. So FC, that's like the bottom part of the Y. That's going to determine which class. Is it IgA? Is it IgD? Is it IgG? Right? Whatever. And then the 2 arms of the Y are the Fab portion. These are going to tell us which specific antigen does this target. And so what do antibodies do? Because we've mentioned antibodies, but like how do they work? We know they target and they help us, help protect us, but how do they do that? Number 1, neutralization. They can bind to the antigen, basically neutralizing the pathogen or the toxin. It stops the pathogen or toxin from getting into host cells. So it's blocking it, right? It's physically making it impossible for that specific pathogen to get into a cell. So that's one thing we can do. We can also use the complement-directed cytotoxicity, right? Basically bind to and activate the complement. Complement proteins come in, they form that complex, they punch holes into the cell bursts because fluid rushes in. There's also antibody-dependent cellular cytotoxicity. The antibody binds, right, to the receptors on natural killer cells, which then helps those natural killer cells or the macrophages kill the cell that is infected or the pathogen. And then opsonization, antibodies can enhance phagocytosis. Basically, it makes phagocytosis easier, right? We tether the antigens to the phagocyte. So basically, like, it helps us catch the pathogen or the antigen so that the cell can easily engulf it and bring it in and destroy it. So let's go ahead and talk about the different classes of antibodies. IgM, this is going to help neutralize pathogens. It can also activate those complement proteins to form that complex. IgG, this is the most common, most abundant. This also can neutralize. It can activate the complement proteins, and then it can also bind to the receptors, right, so that the phagocytes, like a neutrophil or whatever, can easily capsize on or encapsulate that pathogen and destroy it. IgG is also the only one that can cross over the placenta and help protect the fetus and the infant. So it can pass through the placenta, it can pass through breast milk. The IgA, right? This is going to be the second most abundant. This is also going to neutralize, right? But it's also going to secrete across mucosal epithelial. This is also going to help. So this is going to help in the mucosa, and this is also going to pass through breast milk. So this is going to help protect the fetus or not the fetus, the infant as well. The least common is IgE. This is binds to basophils and mast cells, right? This is going to help trigger the release of histamine. So this is going to mediate degranulation, which again is just releasing that histamine and activating those basophils and mast cells. And then IgD, this is going to be expressed on the surface, right, as a BCR. We actually don't 100% know what it does. So interesting, right? So there's some things we don't quite 100% know when it comes to antibodies. So when it comes to where we find them and what they do and whatever and what they look like, let's go ahead and talk about it. So IgM, these guys are big. So we only find them in the plasma and the lymph, right? IgG, we find them in the plasma, the lymph, interstitial fluid, right? They can go from fetus to the placenta, to the baby, go to the fetus through the placenta. IgA, these guys we find in the same locations, but we also find them in mucosal secretion, so in the GI tract, in the respiratory tract, in the urogenital tract, breast milk, and in tears. And then IgE, we find these guys in the interstitial fluid in the skin, as well as in the lamina propria, in the mucosal membranes. Again, these guys, there's not a whole lot of them. It's very uncommon. We generally do not find antibodies in what we call immune-privileged sites. This includes the cerebrospinal fluid, the testes, and the eye. Right? So we have specific barriers that mean that antibodies cannot get through. We don't want to get through to the brain and spinal cord. We want to maintain the blood-brain barrier there. The eye is also considered separate there, and then the testes, because again, the testes were producing sperm that are genetically different. So if you remember from reproductive, we talked about there's a barrier there as well. So opsonization, this is one of the big things with antibodies, right? We can neutralize the antigen, making it inert, so it can't do anything, right? So here we have like an antibody with the binding to a virus. When the antibody binds to the virus can no longer do anything, right? It's neutralized. We can also recruit, we can do complement recruitment. So again, we bind to the bacteria, we then create this chain reaction through complement components, right? And then trigger antibacterial activity, right? Like the complement system, and then punch holes or whatever in the bacterial cell. And then opsonization, right? We bind to that bacterial cell, but then we also bind to the phagocytic cell, which then makes the bacterial cell an easy target, right? It's a sitting duck for our phagocytic cell, right? The phagocytic cell is like, cool, you're stuck. The bacterial cell cannot get away anymore. It's locked down. It's like, chained to the phagocytic cell, and the phagocytic cell can just engulf it and destroy it. Other things that antibodies can do, so we can activate the mast cells and the basophils, and so this is going to trigger the release of histamine. It's also part of the allergy response. So if we are exposed to an allergen, we can then develop allergens or produce allergen-specific IgEs. They bind to the mast cells and basophils. This is what's called priming, and then we get repeated exposure to the same allergen. Sometimes this induces cross-linking of the adjacent IgEs, right? So here, this is what cross-linking is. So here's like a food antigen. It binds to an IgE, and then it binds to another one. So this is what's called cross-linking, right? When we cross-link, right, then we can release pro-inflammatory mediators like histamine, right? Boom. This is what causes the symptoms of an allergy, right? So when you have those allergies, right, this is why your IgE antibodies are binding to your mast cells and basophils, activating them to release things like histamine and cytokines. Other things that we can do, so we can basically go through antibody-dependent cell-mediated cytotoxicity, natural killer cells, monocytes, right? We can use these receptors for IgGs. Basically, IgG will coat the target. It'll bind to an antigen, and then the natural killer cells and monocytes can use that, right, to trigger the granzyme, the perforin. So if we look at the coding, right, here we have IgG, right, the antibody-coated cell, we're binding. Natural killer cell binds to the antibody, giving the natural killer cell an easy way to target that cell that we need to get rid of. Eosinophils, right, can use IgEs that have coded like an anti-helminthic parasite, or a helminthic parasite, so a worm, right, like a worm parasite. They bind to the things like on the worms. Eosinophils then can bind to the IgEs and then kill that parasite, right, by releasing like cytotoxic substances, kill that worm. So in both instances, what we have is we have receptors that bind to the IgG or IgE antibodies and make it easier for the natural killer cell, the monocyte, the eosinophil, to kill its target. Right? That's kind of the general concept here, is with binding to the target that we want to kill, right? The antibodies bind to the target we want to kill, and then the cell that is going to do the killing has an easier time finding and targeting the cell we want to get rid of, or the item, right? The helminth, right? The worm we want to get rid of.